Okay, I only have 5 minutes, but I first want to very sincerely thank the organizers and the audience. This is the best meeting of the year, and I really just love being here and the engagement from everyone, so thank you for that. My name is Brian Chamberlain. I'm a medicinal chemist at TandemAI. We are a drug discovery services company, a CRO, that is dedicated to addressing bottlenecks in drug discovery by combining a state-of-the-art wet lab with advanced computational techniques, be they physics-based or AI. We get very excited when we can combine physics-based and AI approaches to accelerate and overcome these bottlenecks. Last year, I showed a poster in which we showed our implementation of using active learning and RBFE to optimize R groups very efficiently. We and others in the field are doing this, and if you're not doing this, you're missing out on an opportunity to be very efficient and comprehensive in driving the potency of your R groups. In that case, you're able to enumerate large libraries of reasonable compounds using reaction-based framework. Very simple. You know your chemistry. You have a building block library of hundreds of thousands of compounds, and you know you're going to be able to execute on those synthetically. Predicting the potency of those compounds is also amenable to the current methods. You're able to tweak the state of the art for predicting potency, RBFE, in certain ways to make it even more computationally efficient, and then you're able to drive that in an active learning cycle. Again, the synthesis is already baked into how you have set up that experiment, and you're able to execute on those very easily, oftentimes in a plate-based format. Core hopping is an essential activity in drug discovery. I don't think I've worked on a single program in my career where we haven't tried to do a core hop, whether to address a known liability, protect against an unknown liability, protect our IP, or quite frankly, fast follow on someone else's IP. Here, the challenges to this versus R group enumeration are that it's very difficult to enumerate large reasonable libraries of core hops, and then calculating the potency of those is quite challenging. RBFE needs congeneric molecules with small changes to be effective. So probably the most important scientific advance in my poster is our implementation of FEP, which we call CBFE. I won't get into the details now, but it allows you to calculate the potency of compounds in a relativistic framework, purely arbitrary changes. So these compounds you would not be able to calculate using RBFE. This is a BRD4 data set, and we show a very nice correlation here. Unlike an ABFE simulation, you never have to sample the apo state. We apply this method in a soup to nuts prospective case study on SOS1 inhibitor MRTX0902. And for me as a medicinal chemist, the most interesting aspect of this work is the enumeration. We compare multiple enumeration methods, sort of the most common ones in the field, link invent, both the prior model and reinforcement learning on basic objective endpoints, CREM and RECORE. We did a lot of work to get those libraries for those methods, if you're aware of them, to work on a core hop and give us a meaningful number of compounds, and we compare and contrast those. We also have a human medicinal chemist design the compounds as a human medicinal chemist would by looking at the crystal structure and thinking about what types of reactions are expected to perform well. A lot I can say about the enumeration of all those different modules, but the two things I'd like to highlight is that generative AI is the most important computational approach to generating core hop libraries, simply because of its ability to generate very large libraries, which you will not be able to access with library-based methods. Very interestingly to me, if you compare the compounds enumerated from the generative AI approach and look for the human design compound, you can't find it. Even if you were to run link invent with reinforcement learning onto a very narrow molecular weight around which the molecular weight we're looking for, you would have to enumerate maybe 100 million compounds to maybe enumerate the compound that a medicinal chemist came up with. So I think that's really important. We have to combine and take the best features of all of these different enumeration techniques to achieve our goal. We then dock all these compounds using a few algorithms to make sure whatever we're observing is not just strictly related to VINA or RDoc or whatever. And I have trends that I can show you. Probably the most interesting one for the next 15 seconds is that the library-based methods are really perform well at reproducing the RMSD pose of the reference ligand, and that's super important to being able to deploy the physics-based methods. So I take it all the way. We run the CBFE. I have the real data on how that performed. So you can assess for yourself if you think it's interesting or if you would want to use that tool. And then I show the actual IC50s. I don't have time now, but I can kind of philosophize how these techniques are impacting me as a medicinal chemist. Of course, none of this really existed, or if it existed, it wasn't easy to use, or someone like me to access and deploy without really a lot of special computational help. So 5 minutes, exactly. Thank you.